Hi, this is Maggie Fires, and this is my Chapter 16 presentation. So, this presentation is about the third line of defense, which is called adaptive immunity. And this line kicks in when the first two lines will fail, and like your physical barriers and innate responses aren't enough. And what's unique about this is that it's highly specific and particular to a certain pathogen, meaning it targets one invader at a time. And it also develops a memory. So after you recover from something like the chickenpox or another virus, your immune system will remember that virus and will attack it more effectively if it ever returns. So the major players in this are B and T lymphocytes, and they're special because they recognize the foreign markers called antigens. And adaptive immunity is it's something we acquire over time. So like innate defenses, they're always on, but adaptive immunity develops over time either through natural infections or vaccinations. So there are 4 stages of adaptive immunity. It doesn't all happen at once. So it starts with lymphocyte development and differentiation. So this is where stem cells mature into B or T cells, and B cells will finish maturing in the bone marrow, and T cells finish maturing in the thymus. And then after that, we move on to antigen presentation, and this is handled by the dendritic cells and macrophages, and they basically show the invader to the T cells so that the immune system knows what to attack. And then we have activation and clonal expansion. This is where B and T cells will multiply into like huge armies of identical cells, and some of these will become memory cells, which will stay behind for long-term protection. And then we have the effector phase. So this is when B cells will produce antibodies that will circulate and neutralize pathogens, while the T cells will perform cell-mediated immunity, and this destroys infected cells directly. And I don't think that was meant to be a step 5, but these 4 steps together form up the adaptive immune responses, and they're powerful and specific. So in the immune system, the immune cells recognize each other and communicate through each other, and they do this through markers. So markers are proteins that are on the surface of cells, and they let the immune system tell its self from not its non-self. So there are 4 different functions. First, they attach to foreign antigens, and then they bind to self markers like MHC molecules to avoid attacking their own cells. And then third, they transmit signals to coordinate immune responses. And then fourth, they help in cellular development. So they guide cells to mature and perform their specific roles. So then another type of marker is called CD molecules, and these are important for identifying the different immune cells because there are like almost 400 of them, but there are some that are well known like the CD4 and CD8, which help distinguish the different type of T cells, but then there are so many other ones. So these surface markers are like communication tags. They help identify and ensure that the immune reactions will stay organized and targeted. So now I'm going to talk about the major histocompatibility complex, and this is also called MHC. This is a gene family that codes for proteins that will be found on the cell surfaces. So these proteins are essential for helping the immune system tell the difference between self and non-self. And you'll find them on almost all cells except for red blood cells when the immune system checks for the markers. And it makes sure that the cells belong to you and not something else. So if it detects something different, like a foreign MHC, then it'll trigger an immune response. And there are 3 different classes of MHC genes. So there's class 1, class 2, and class 3. So class 1 molecules, these are on all nucleated cells. They display normal self-markers. But then class 2 MHC molecules are on macrophages, dendritic cells, B cells, and they're used to present antigens to T cells, basically showing them what to attack. And then class 3 MHC genes, these produce proteins that are involved in the complement system. These help destroy pathogens. And so now we're going to talk about antigen recognition. So this is how B and T cells work in different ways. So B cells have receptors that can bind directly to antigens, and they float freely in the body. And then we have T cells, on the other hand. They require antigens to be processed and then presented by another cell. So usually this is with an MHC molecule before they can recognize it. So the cells that perform this task are called antigen-presenting cells, and these include macrophages, dendritic cells, and B cells. So once a B or T cell recognizes a specific antigen, then it undergoes clonal selection. So clonal selection means it multiplies into identical copies, all with the same receptor for that antigen. And then from there, the B cells can develop into plasma cells and release antibodies, while T cells can differentiate into helper or regulatory or cytotoxic forms, and these handle cell-mediated responses. So this system allows the immune system to respond and be targeted and efficient and help the body clear pathogens, and then also remember it for future defenses. So we talked about maturation of T cells and B cells. They both go through the maturation process, and it gives them their specific roles. T cells mature in the thymus, and this is where hormones guide their development. Hormones guide their development, and they gain 2 important identifiers. So this is CD3, which helps with receptor binding, and either CD4 or CD8 co-receptors. So CD4 is mostly found on T helper cells, which work with the MHC class 2 molecules, and then we have CD8, which is found on cytotoxic T cells, and which these bind to MHC class 1. And then once they're mature, the T cells will circulate in the lymphatic system and complementary circulatory systems and constantly monitor for infection. So on the other hand, B cells, they mature in the bone marrow and develop immunoglobulin receptors, or Ig, on their surfaces. So these receptors help them recognize specific antigens later on, and after they mature, both B and T cells move into secondary lymphoid organs, and this is like the spleen of lymph nodes, and they'll eventually encounter pathogens. So generating immense lymphocyte diversity. So each lymphocyte, they will have unique receptors. So this happens through genetic recombination. So this is where pieces of DNA in developing B and T lymphocyte cells are arranged, and they will make new antigen receptor genes. So there's over 500 gene segments that can recombine, and this means your immune system can potentially produce like over 1 million types of combinations. So that's how every single lymphocyte can recognize one specific target. And then this all happens before an infection occurs. So like during fetal development, these receptors are built and expressed on the surface of lymphocytes, but each cell becomes a clone representing a single antigen. So now I'm going to go a little bit more into clonal selection and deletion. So these are 2 processes that happen during lymphocyte development. So clonal deletion, this happens first. It eliminates any lymphocytes that might attack the body's own cells, and ensures immune tolerance, meaning the immune system learns not to react to self-antigens. And without this step, autoimmunity could occur where the immune system mistakenly destroys healthy tissue. So then there is clonal selection. So this occurs after an antigen has been exposed. So when a pathogen enters the body, only the lymphocyte with the right receptor for that antigen is activated, and the cell divides repeatedly in a process called mitosis, which you've heard about, and this produces many identical copies or clones. So some of those become effector cells, and then others become memory cells that remain for future responses. So this system helps keep the cell safe and not destroy any healthy tissue, but also prepares for foreign invaders. So now I'm going to talk about the B cell receptor structure, or the BCR. So this is an immunoglobulin molecule, and it has the same kind of structure that becomes an antibody once it's secreted, and it's made up of two identical chains, so two identical heavy chains and two identical light chains, and they are all linked by disulfide bonds, and they form this Y-shaped structure. And then at the tips of the Y are the antigen binding sites, which are the variable regions, and these determine what antigens the B cell can recognize. And then the variable regions, these are incredibly diverse because they have genetic rearrangement that happened during the B cell development. And then each receptor fits one specific antigen. So this gives the molecule shape and stability, but once activated, the B cell can release these immunoglobulins as antibodies, and this allows the immune system to neutralize pathogens. So then we have the T cell receptor structure, or TCR. So this is related to the B cell receptor, but it functions differently. So B cell receptors and T cell receptors are made through DNA rearrangement, and they both have variable and constant regions. But structurally, they consist of the two parallel polypeptide chains, and their job is to recognize process antigens that are displayed by the MHC molecules on the surfaces of the APCs. So the big difference is that T cell receptors are never secreted, so they stay anchored in the T cell membrane, and this makes sense because T cells don't release antibodies, and instead they interact directly with other cells. So this structure gives it high specificity, and each T cell responds to only one antigen MHC combination. So now I'm going to talk about the characteristics of strong antigens. So the immune system is wired to recognize things that are foreign. So molecules, microbes, animal cells, or even humans are often seen as threats too. So the more complex the molecule, the stronger the immune response is and can provoke. So proteins and glycoproteins, these make excellent antigens because they have varied and intricate structures that immune cells can easily detect. So several chemical groups will serve as antigens, like proteins, lipoproteins, glycoproteins, nucleoproteins, and more. These are highly immunogenic, and the size of them matters too. So a small molecule that repeats the same unit, like glycogen, won't normally be recognized as antigenic, but a smaller, more complex molecule like insulin can be. So now I'm going to talk about epitopes and haptens. So lymphocytes don't recognize an entire antigen molecule, they recognize specific parts of it. So these small distinct regions are called epitopes or antigenic determinants. So each epitope is the portion of the molecule that directly interacts with the immune receptors on B or T cells. And then there are haptens. So these small molecules, they can't trigger an immune response by themselves, so they're incomplete, and they have to have a large carrier molecule like a serum protein, and it becomes large and it becomes complex enough to be recognized as an antigen. So this is why some drugs or chemicals can trigger allergic or immune reactions, and they act as haptens, so they bind to proteins in the body and form a complete antigen. So now I'm going to talk about alloantigens and superantigens. So alloantigens, these are cell surface markers, and they vary among species, and because their markers are unique, they become important in medical contexts, so like blood transfusions, organs transplants. And then superantigens, these are microbial molecules. They massively overstimulate the immune system, and they activate T cells at levels way higher than normal antigens. So this is why they are superantigens, and this overactivation leads to huge amounts of cytokine